Well, hello, everyone. Thank you very much to IDT. Feel very honored to win the award this year, and very honored to be with you all tonight. My background, I'm actually a cave biologist and molecular ecologist. I've worked on a variety of different organisms. In particular, I work on things that live in caves and in karst environments. So karst in particular is a terrestrial landform composed primarily of soluble bedrock, so things like limestone, carbonates, marble. So there's a variety of resources that caves and karsts provide. Obviously, some of the most obvious are groundwater, so for drinking water, for irrigation, mineral resources from mining, for limestone itself, for guano, for saltpeter, particularly from a historical perspective. But what I primarily work with are the metazoans, the animal life. So an avenue of research that we've starting to explore now is using environmental DNA to actually try to better understand the distributions of many of these species. So essentially, as we're sitting in this room right now, we're all constantly shedding DNA into that environment. Well, so are the animals that are living in their habitats. So the general idea is, we can go out, I can go to a spring, I can go to a cave, I can collect a water sample, I can then bring that back into the lab, extract the DNA, and then using, IDT developed primers, we can then screen for different species, develop assays for different species to see if they're present or absent within a water sample from a particular site. Some of the values of DNA are that, it can be oftentimes, at least in terms for cave biology, better than traditional survey methods. So it could be essential for, long-term measuring. It's relatively cheap. So as an application for this, so our first kind of preliminary foray, we used eDNA to try to detect an endangered groundwater isopod living in the Washington, D.C. area. So it lives in this very specialized hypotelomenoreic habitat, so these kind of groundwater seeps, difficult to sample. So we collected water samples from the field, from a variety of different sites, and then developed TACMan qPCR assays using IDT's primer quest tool, tested these primers and probes in the lab before applying them to our field samples. And kind of the punchline here is that we were actually able to detect this species at 4 sites, including 1 new site, which is really, really encouraging. And we're now using this approach for many other species, and that's going to be the focus of this proposal project. Again, I want to thank IDT for helping support our work.